And so I just wanted to say too, so the full videos of this will be on OHBM on demand afterwards. I'll also try to put these, so you'll be able to watch them again. I'll also try to put the slides up on my website, and they'll also be on the OHBM website, so everybody will have access. And hopefully we'll have a few minutes at the end to have the panel up here, and everybody can ask questions about anything that they're interested in. Okay, so last talk of the day is after the association, functional and biological validation of variants. So here you see the full spectrum of what we went over today. We're really hitting just small parts of each part of the imaging genetics field or the genetics field in general. So hopefully it gives you just a taste and points you in the right directions of where to go. So here we're going to go after you've conducted all of these tests to try to identify either genetic variants or genes in the genome. Where do you go to next? What do you try to do to understand the biology better? Okay, so I'm going to focus here on genome-wide association studies, and in those we do, as Derek and others have talked about, we profile the entire genome for common variants, and we test each of those common variants associations with a particular trait, in this case putamen. We can then plot them in a Manhattan plot shown here, where the y-axis is the significance, the association of each of these individual SNPs, which is a dot, to phenotype we're interested in, which is in this case putamen. So we found a hit, so we found something that's genome-wide significant, survives all of our strict genome-wide significant thresholds. So there's a difference between what we have found and what we actually want to know. So what we have found is that variation in a locus of the genome, which significantly influences your trait, which can be brain structure or disease. But what we actually want to know is something much deeper. So we want to really understand a mechanism by which genetic variation influences brain structure or function or disease risk. A locus is actually a bunch of correlated SNPs or a bunch of correlated genetic variants that are inherited together. We can't really pinpoint the causal genetic variants using just genome-wide association alone. So there's tools to prioritize those. There's tools to try to move from genetic variants to gene of action, to move from gene of action to biological pathway of action, and to move, since we're studying brain regions, we can actually look to see if a genetic variant is affecting a particular brain region or a wide amount of brain regions. Okay, so I'm going to try to teach you some tools that you can use to study these things, but first, a word of caution. So it's very easy to come up with a story by which your gene that you have identified can explain whatever trait you're interested in. So there's a lot of studies, a lot of just warnings in the literature, and I just want to point you guys towards some of these warnings. So one says, human genomes have a high level of narrative potential to provide compelling but statistically poorly justified connections between mutations and phenotypes. Another one, a critical challenge for biologists will be avoiding premature hypotheses born of biological plausibility and just so stories. And finally, findings from single association studies constitute tentative knowledge and must be interpreted with exceptional caution. So this is just to say biological plausibility is not a substitute for statistical significance and is not a substitute for experimental validation. So be careful, but having said that, let's go and explore some tools. Okay, so how do we explore biological mechanisms? So I'm going to go through different things. First is exploring the associated locus using some visualization tools. Okay, so there's many different visualization tools for looking at the genome. Those genome tools can be classified either as dynamic, so it's easy to sort of move around in them, or publication quality static images, which are useful if you're trying to publish a paper. I'm going to focus on the dynamic ones, and specifically the UCSC genome browser, which is the most widely used genetic tool, basically the most widely used bioinformatic tool that exists. It's fantastic, and everyone should be aware of it. So I'm going to go over just briefly how to use it and where you can find it. So there's some advantages and disadvantages of each of these different tools. And I have, at least for the end, I have at the end of my slides some code that you can use to run GViz, which is a way of plotting any kind of genomic regions you're interested in. Okay, so here's the UCSC Genome Browser. This is a website. If you learn one thing from this talk, it should be this website. This is a very important website. So the first thing that you can do is just go select your species of interest, which in our case mostly is human, because it's an organization for human brain mapping. You can select your assembly. That's the coordinate system. So the genome is constantly updated, and the base pairs that identify sort of the map of those base pairs is constantly updated. So the coordinate system from the 2009 version of the genome is not the same as the coordinate system from the 2016 version of the genome. So you just have to be careful which one you're using to make sure you're not going between different versions. And there are conversions between the two, but just be careful what you're using. So then you can go in and type a gene or a SNP of interest. This is a SNP. This is the one associated with putamen, so you can type that in. And here's what the plots look like afterwards. So this is a plot where we're exploring the locus, the region of the genome near this particular SNP, near this one location in the genome. So the location in the genome, here is where the variant is. This is the variant associated. And then I zoomed out, and you can see that I zoomed out to 50 megabases. So this is like a huge region, millions of base pairs that you can see here. And luckily, because biology has been working on the genome forever, not forever, for a long time, we have identified regions of the genome where we know the functionality. We don't know the functionality of most of the genome, but we know some functionality for some regions of the genome. So one of those pieces of functionality is genes, right? Genes can encode proteins. So for example, this is KTN1 is a gene that encodes a protein. And there are multiple isoforms of that gene that exist, so multiple different forms of that gene that exist. There's also other types of genes, like long intergenic non-coding RNAs, the link RNAs. Those do not encode for proteins, but do encode for some sort of RNA that's expressed in the genome. Many different types of RNA that are expressed in the genome. So each of these represents a different track, and because we have different annotations measured in different ways, in different cell types, you can display those tracks on top of the tracks, on top of the region that you're interested in this case, this variant. So this is a great tool because it takes all of what we've learned about biology and sort of puts it into one unified map that we can explore. Okay, so as David talked about, there's many different types of genetic variation that exist in the genome. Some of that is extremely interpretable. When we know the codon code of the genome, we know the 3 base pair code that turns the genome, the DNA, into proteins, right? And we know that if we mess up some of that genome code, how it will change the proteins. So those changes can be, for example, missense mutations. Missense mutations can change the protein coding structure of the genome. So instead of one amino acid, you're going to get another amino acid. We can have insertions or deletions. Those are small little places where we delete a region of the genome or insert a new base pair. And we can also have nonsense mutations. Those can make a stop codon, a premature stop codon that results in the protein not being fully expressed. We can also have splice site mutations that alter the splicing, those different exons, splicing of the different exons together. And we can have intergenic or intronic mutations. And so just to give you an idea, about 3% of the genome encodes for proteins. The rest, the 97% of the genome, is intergenic or intronic. So most of our GWAS hits, 88% of significant genome-wide association hits, are found in intergenic or intronic region with no clear gene of action. So this is why GWAS is so complicated, because we do not really understand what the regulatory, predicted regulatory function of these variants are. And just to give you an idea, too, of how complicated this is, because we're not just looking at one genetic variant, we're looking at a bunch of inherited genetic variants that are correlated together. GWAS loci tag very large regions with multiple different functional elements contained within. So for the schizophrenia GWAS that David was talking about, the max size of the LD block was 800 kilobases. That's 800,000 bases. The schizophrenia GWAS, the mean size was 171 kilobases. And just to give you an idea, the mean gene size is 29 kilobases. So even though you're found a significant locus, that locus may encompass many different genes. So it's not easy to move from a genetic variant to a gene. So rare variants, including missense or nonsense mutations, have a very clear gene of action, but they're rare, so they're harder to find. So. Okay, so one way to identify the possible coding functionality of a locus is to use the linkage disequilibrium, use the idea that a lot of genetic variants are inherited together, and then try to see if a genetic variant is often inherited with a variant of known function. So if a genetic variant that you find that's most significantly associated is in LD, so it's highly correlated with a missense SNP, we might use that to predict the functionality. So this may be affecting gene A because it's a missense SNP in gene A. From our experience in the Enigma Consortium, this is extremely rare to find. You don't usually get this lucky. But you always want to check this first because this is a very easily interpretable functionality. So to do this, you can use 1,000 genomes for the LD for determining the correlation between different genetic variants, and then use the UCSC genome browser to determine the functionality. And I have some slides at the end of this that give you specifically how to do that as well. Okay, so I'm going to go over a little bit of epigenetics too, but using epigenetics as annotation, not really using epigenetics as the EWAS as Sylvain described. Okay, so when genetic variants fall in intergenic or intronic regions, we often assume that they have a regulatory potential, that they're affecting the regulation of gene expression. Okay, so how does gene expression regulation canonically work? Well, so here you kind of have it all in one picture. So you have DNA, which is this little strand here that's wrapped around histones, which are these little things, and the histones have tails. Those tails can be methylated or acetylated. There's certain regions of the genome that are called heterochromatin, where the DNA is tightly wrapped around those histones. And so DNA binding proteins can't get in there and do any kind of action. There's certain regions of the genome where DNA is open and accessible to transcription factors or DNA binding proteins, and therefore allows them to function in whatever cell type you're interested in. You have regions of the genome that are transcription start sites where RNA polymerase can bind, and those transcription start sites can be influenced by distal repressors through the genome sort of folding onto itself like this. So if, for example, this a transcription factor binds to this distal enhancer, that distal enhancer physically interacts with the transcription start site and the RNA polymerase at the transcription start site, it can increase the expression of a particular gene. Okay? So there's various ways to measure all kinds of epigenetic features. You can measure DNA methylation, as Sylvain talked about. You can measure chromatin interaction, that's the touching between two pieces of chromatin. You can measure protein-DNA interaction if a DNA binding protein is actually bound to a particular region of the DNA. You can measure chromatin accessibility, so the hypothesis there is that chromatin is more open or accessible in regions where it's functional, where it's actively functional, even if those regions are not coding for proteins. And there's a cool new field of biology called enhancer RNAs. So those enhancer RNAs are where enhancers are, there seems to be some actual transcription of the enhancer element here that's separate from the promoter element here. Okay, so here's what these all look like. So in the end, what we want is an annotation of different regions of the genome. And again, we know those exon regions pretty well, but now we want annotations of these non-coding elements, these enhancers, insulators, and repressed elements. So what we can do is measure all of these different factors in different cell types and try to see if we can annotate in different cell types what the functionality of the region is. So we can measure RNA, and we can see RNA is expressed both at the enhancer element and at the exons. Excuse me. We can measure transcription factor binding using ChIP-seq, that's chromatin immunoprecipitation. We can measure things about the histone tails, and those tell us, those are often associated with either repressed chromatin in this histone 3 lysine 27 trimethylation, or can be associated with active enhancers, histone 3 lysine 27 acetylation. And we can also measure things like Hi-C, which I'll talk about a little bit more, which tells us if two regions of the genome are actually touching and interacting together. So all of these different annotations, you get all of these different tracks for each different cell. That can tell you something about the functionality of these elements in different cell types. Luckily, you don't have to do this all for yourself because NIH funded a giant project called Roadmap Epigenetics. to do this in many different tissue types at different periods during development. So you have both adult and fetal development, and you have different regions of the brain, interesting for us, that were profiled through a variety of those different epigenetic annotations. There's also the ENCODE consortium, which I'm not going to talk about here in detail, but the ENCODE consortium profiled a bunch of cell lines. So those are a little bit less useful for interpretation in things that we do, more useful for things like cancer. Okay, so how can epigenetic annotations help us with our interpretation? They can allow inference of tissue or cell type affected by looking at enrichment of epigenetic marks in regions of the genome that were associated with our trait. I'll give you one example of that. So there's also inference of developmental time period, again through enrichment. So if these genetic variants that we find seem to be enriched in fetal enhancers but not adult enhancers, that can tell us that maybe the time period of action was in fetal development. Since we have those big loci that are hundreds of kilobases long, it can help us prioritize functional variants within those large associated loci, since we know something about the functionality of those. And it can give us ideas as to the mechanism of individually associated loci. So is it an enhancer, an insulator, or other type of regulatory elements? And so this is a complicated slide, but from the Epigenetics Roadmap Consortium. So I know you can't see it, but just to give you an idea of how these enrichment type of things work. So on the left-hand side here, you can see genome-wide associations done from many different types of traits. So there's been genome-wide associations from, for example, rheumatoid arthritis, multiple sclerosis, all kinds of cholesterol traits. And then you can see DNA's hypersensitivity evaluated in many different cell types from the epigenetics roadmap at the top here. And then for each of these comparisons, you can look at an enrichment to try to see if genome-wide association hits often fall within DNA's hypersensitivity regions of a particular cell type, okay? And so that can give you some interesting insights. So even though I can't read it too, I think this one is multiple sclerosis. So multiple sclerosis seems to be enriched in immune cell DNA's hypersensitivity sites. Rheumatoid arthritis also seems to be enriched in immune cell DNA's hypersensitivity sites. So these aren't necessarily surprising, but do give you insight as to a potential mechanism by which these genetic variants are having their effects. An interesting one is that irritable bowel disease down here seems to also be enriched in immune cell types, which is interesting. And this one here, so all of these are cholesterol traits, like LDL cholesterol levels, for example, HDL cholesterol levels, and they seem to be enriched in liver DNA's hypersensitivity sites. So for brain, this can also give you some ideas and has been applied also in like schizophrenia to give you ideas of the region of action. Okay, so David talked about this a little. When oftentimes we have this locus, the locus is far away from a gene. How do we move from the locus to a gene of action? So there's two ways I'm going to talk about doing this. One is expression, QTLs, and the other is chromatin interaction. Okay, so here's the problem again. So we have a genetic variant that we found, or a genetic locus that we found. It's near multiple different genes. How do we move from a genetic variant to the gene of action? So we can do this in multiple different ways. One is by doing EQTL, expression quantitative trait loci studies. So in this, we can see that the red variant seems to be associated with higher levels of expression of gene B, whereas the black variant here seems to be associated with lower levels of expression of gene B. However, that red variant doesn't seem to affect at all expression of gene A. So from this, we can maybe say that we can suggest gene B as a possible gene of action for this genetic variant. Okay, so I'll give you a little bit more about this, like a biological interpretation of this. So imagine you have, let's say, a bad variant. That bad variant is indicated by this star here. That's a change in the actual base pair of DNA. That bad variant allows the transcription factor to bind, so maybe creates a new transcription factor binding site, allowing transcription factor to bind, creating higher levels of expression of a particular gene, creating higher levels of protein, and therefore influencing disease risk. Okay? Now let's say another person has a good variant. That good variant is associated with methylation at that region. The methylation prevents the transcription factor from binding, therefore decreases the levels of this risk gene. You have less expression of the risk gene, less protein, and therefore a lower disease risk. So this is like a biological, this would be like a full biological cascade, and we know this for very few traits. A full biological cascade to understand what the mechanism is behind an EQTL association or a disease QTL association. Okay, so in EQTL literature, it's important to know the difference between cis versus trans and local versus distant. And these two are often conflated in the literature, so it's important to know what the differences are. Local just means close. The genetic variant is close to the gene of action. That's it. Distal is far away. Local does not necessarily mean cis, and the same. Distant does not necessarily mean trans. So in a local change, which is called a local cis regulatory element, you can have a genetic variant, and that genetic variant can, for example, alter transcription factor binding, which then leads to decreased or increased expression of a particular gene. So importantly, that happens on the same chromosome. So the same strand of DNA has the genetic variant and has the alterations in expression. Trans, on the other hand, is, for example, if you have a genetic variant affecting the expression of a transcription factor, for example, it will affect many different genes, so can affect genes across the genome, and will importantly affect both strands. So it doesn't just affect the one strand, as in the cis, but affects both strands, because you have that diffusible factor that can bind to both of those different strands. Okay, so things to be aware of. In the literature, often studies proclaim a cis-EQTL was conducted when they actually conducted a local EQTL, looking in ±1 megabase from the gene of action. So they assume a cis mechanism, just beware the conflation. It's very consistently used in the literature, but it's not exactly correct. Most published EQTL studies are local EQTL studies for power reasons. Distant EQTL studies measure all genes by all SNPs. So you have, let's say, like 6 million SNPs that you study, and you have 20,000 genes, so 6 million times 20,000 is a big number. Whereas if you study local EQTL studies, you look at only the SNPs close to the gene. So you dramatically reduce the number of multiple comparisons that you're doing. So why does cis versus trans mechanism matter? So it will tell you if there's direct or indirect causation, right? So is there a direct influence that you're looking at, or is it mediated by some other factor, some other diffusible factor in the system? If you just want to know the gene of action, it's not super important. Okay, so EQTLs allow mapping of a variant to a gene. So we have a SNP associated with a trait. We can run an EQTL and just see if any of the genes seem to be influenced by the SNP. And here's an example of what an EQTL looks like. So here we see genotypes on the x-axis at the bottom and the expression of a particular gene. In this case, it's that KTN1 gene that I showed earlier. And so it seems that the SNP that's associated with putamen volume actually is also associated with the expression of this gene, KTN1. So we think we suggest a gene of action for this trait. There's many EQTL databases that exist. There's many more that are coming along. There will soon, I imagine, be cell type specific EQTLs, some of which we're working on in my lab. So here's just some websites that you guys can look at to be sure you have. And yeah, all the slides will be available too, so don't worry. Just one point about the genome being complicated and much more complicated than you want it to be. Just because you have a genetic variant influencing, for example, chromatin accessibility, does not mean that genetic variant influencing chromatin accessibility also influences the expression of the gene. Just because you have a genetic variant influencing the expression of a gene doesn't mean that it also influences the protein, the steady state protein levels. So biology is complicated, and so you basically have to test it at multiple different levels of biology in multiple different cell types, which is very expensive and difficult. Okay, so next thing I'm going to talk about is chromatin interaction. That's the actual touching of two segments of DNA. So the touching here. So this is measured through a procedure called Hi-C. In this procedure, you have your intact nucleus. That intact nucleus has a bunch of proteins that are in there, and you can fix those proteins. And so any two regions of the genome that are touching each other, they'll be still close to each other because they're fixed by these proteins. Then you can sort of just cut those and tag the ends of them together, and then sequence the ends. And by sequencing the ends, you're going to be able to find 2 places in the genome that were actually touching each other. So just to show you an example of this from non-neuroscience literature, this is a famous gene called FTO. And there's genetic association. It was one of the first genome-wide significant genetic associations that was detected, was variation in the FTO gene affects obesity and body mass index. And so people thought, oh, FTO is the gene of interest. FTO is what we should study, and we should spend a lot of money on it. But it turns out that using both chromatin interaction and EQTL studies, that genetic variation in this locus is not actually associated with differences in FTO gene expression. It's associated with expression in IRX3 and IRX5, which are these genes downstream. So this is just an interesting example to make sure that you don't necessarily just pick the closest gene. Okay, and here's one example that we worked on to explain schizophrenia genome-wide association hits. So schizophrenia genome-wide association hits are often, again, in intergenic regions. And here we did Hi-C, so chromatin interactions, trying to look if we can explain some of the genes that are involved with risk for schizophrenia. So in this case, you can see here the genes on the top. Here are prioritized SNPs that we think could be the causal SNPs in the region. And in fetal brain tissue, we were able to profile to see if the region that's associated with these SNP is actually physically interacting with a region at the transcription start site of different genes. So here we can see that these SNPs, which are creating risk for schizophrenia, are actually physically touching DRD2. So this is, we think, an important example because these SNPs, although they were close to DRD2, didn't really have any evidence for impacting DRD2. But now we show that they're actually physically touching, which is some evidence for that. So as with any technique, there's a lot of caveats. So chromatin interaction does not guarantee functionality. Genome scaffolding within the nucleus is likely a major driver of chromatin interaction that may not directly lead to function. So things are just sort of stuck in the genome. So it's not necessary that just because 2 things are touching that they're necessarily functional with each other. Chromatin interaction is a pretty new field. So chromatin interaction is measured in the large bins. So highest resolution so far is around 10 kb. So it can't point us to an individual base pair of interaction. So even though that SNP is really what we want to study, we study 10 kb around that SNP. Also, it has not yet, since it's very, very expensive, usually you have to do whole flow cells, which are like tens of thousands of dollars to get enough data for this. It's not been measured in a large amount of tissue or cell types yet. Okay, so I'm running a little bit out of time. So again, these slides will be available to you, so I'm going to skip pathway analysis, and I'm going to go just quickly into what experimental manipulations we can use. So there are many model systems for exploring biological mechanisms, right? So those can be humans, right? So we do human imaging, that's one. But you can prioritize each of these different model systems along 3 different axes. So one of those axes is human relevance. So obviously humans are very human relevant. Mechanistic insight. So unfortunately, we can't modify or do a lot of really, especially genetic manipulation experiments in the human. Humans have low mechanistic insight and throughput. So how quickly can we do these experiments? So there's different model systems for doing different things. We can use mouse models, which have high mechanistic insight. We can modify their genetics, low human relevance, they're not human, and pretty low throughput. There's also induced pluripotent stem cells and primary human neural progenitor cells derived from fetal tissue. These are pretty high on all 3 axes. They're probably a little bit lower throughput than this sort of older review lets on, but they're highly human relevant and they have high mechanistic insight. We can modify their genetics and do experiments on them. So neural stem cells can be derived from embryonic stem cells, induced pluripotent stem cells, or fetal tissue. They're one of the few ways that we can study the living cells of the human brain. We can genetically modify them to be similar to patients with a disease or take cells from a diseased patient, and they can theoretically be used to screen drugs at high throughput. And without going into the ways that you can derive them, there's also this new technology called genome editing, which is sort of evolved from different types of genome editing, but this will really allow us to test the causality of each of the different loci that we're studying. So one of the newest versions of genome editing is called CRISPR-Cas9. In this, there's a protein called Cas9 that can be guided to anywhere in the genome that we want with very little restrictions using guide RNAs that we can design. And these can be used to actually modify the genetics to either create large frameshift mutations or indels, or to actually, at very low efficiency currently, to actually insert base pairs of interest. So there's only been, I think, one instance of this being used, as far to my knowledge, in neuroscience so far, which is for Parkinson's disease, where they've actually gone into neural stem cells, previous they did a genome-wide association, found a genetic variant of interest, prioritized to find exactly which variant they wanted to test, found some got some neural stem cells from a particular donor, had one base pair, modified that one base pair to change it, and basically did the exact experiment you would want to do. So you have an identical twin, but only changed by one base pair. And they tried to look at what the biological mechanisms for that Parkinson's disease associated variant was. So that's a fascinating study, and that's where the whole field will try to go, is to try to actually do experimental modifications using neural stem cells or other types of model systems. Okay, so last slide. So identifying genetic loci is a causal foothold into understanding novel biological mechanisms. It really doesn't point us to novel biological mechanisms on its own. It's a foothold for understanding that. There are many databases and tools that will allow you to form hypotheses about biological mechanisms. Hypotheses can be tested using model systems and genome editing. And just again, a word of caution, it's easy to make a story. Try to let the evidence guide you for what you're doing. Okay, thank you guys very much. So I know that we have about 5 minutes left in the session. I'd like to call the speakers to come back up here. And if anyone has questions for us, we would love to answer your questions. We're happy to stay a little bit later too to answer any questions you may have. And if you guys don't ask questions, then I'll ask questions to them. Oh, do you guys want to? Yeah.
I was just wondering if you were aware of any resources coming out that would give cell-specific expression profiles in early human brain. Is that on the horizon?
So there are a few resources to give cell type specific expression profiles. The Allen Institute is one that's definitely doing this in mouse brain and human brain. There's DropSeq profiles that are being generated by Steve McCarroll's lab and Aviv Regev that are, I think, out for both human adult brain, and I know they're being generated also for human fetal brain as well. So there's also cell type specific EQTL studies that are starting to happen, I think, and I think those will come out decently soon too. Like our lab's working on them, and I know other labs are working on them as well. So.
Is this working?
Yep, we can hear you.
Yeah, thanks for a great session. I actually have a question that's picking up on something that JB mentioned in his talk, which is, I don't know how relevant that is to people here, but there might be some people here who are looking into genetic variants that are related to schizophrenia or Alzheimer's, and we're doing quite small studies that people have like 20 people per group or maybe 30 or 50. And I'm actually wondering what your suggestions are to people who work in students and postdocs. Is there any point to doing these smaller scale studies, or should we nowadays just look at large studies such as biobank data and these kind of things to get an idea of what a specific gene or gene variant is actually doing in terms of I guess we all have an idea of problems with replication and these kind of things.
So I guess the if you have a very strong hypothesis, if you know exactly the very precise question you want to ask, if you pre-register that question, there's a good validity in doing those small studies, because you're just trying to confirm that result. And the other thing is that even small studies, if we could accumulate the results of and make those available and spread those things, we could then start to do meta-analyses that are reasonable. But it is true, I still do think that we do have to think differently in terms of how we do the science now. We do have to think in terms of making sure that part of our job is to make sure that the data are accessible and documented, such that all those studies can be. But it's really like a shift of mind in the scientist. Part of the job is not to, the job is not only to get to exciting results and publish in high-profile journals and whatever. I think that was you, right? Unfortunately, it is actually in the back of the mind, even if it's not completely explicit, it is the definition of a scientist's job at the moment, that is to publish things. So I think we should publish data, and publish data means documenting data, making sure that they are accessible. I don't mean to make them public, because sometimes they are private and delicate information to be handled. So I don't mean that, but I mean UK Biobank style, HCP style, where you do have access to the data. You do have to sign on and making sure that you're doing legitimate studies with those things. But it is making the data available, and therefore you could start to sort of consolidate and agglomerate data and results.
So I'll add on to that just briefly, which is there have been a lot of resources that have been talked about today in this session, different resources that are available, different models that are apparent. So even if you're doing a really small study looking at a particular variant, let's say, and some trait, it's quite possible that similar data exists in a freely available resource, and that you can look to try to replicate what you've done in a different data set. And that pushes the utility of your paper or your experiment significantly higher. So I would highly recommend just looking around once you've got some piece of data. They may not have the same exact phenotype you have. As a matter of fact, they probably don't. But they may have something similar enough that you can ask a global kind of question, or you can use some other data to rule out a different possible answer to the one that was the most simplistic one. And those are the kinds of things that you're starting to see a lot more of now that there's more and more data available. And you want to, and it's an important point, right? Because even if you're in the one postdoc in Antarctica, you can still download the ADNI data and ask the same question, right?
Next question.
I have a question for Menon. As a person who works with imaging, like you said, I've seen a couple of papers trying to relate, like the Alan Brand gene expression with imaging, like better. I would like to know how cautious, like if I do an analysis like that, how cautious the interpretation should be, considering the sample are from entirely different, the gene expression is from a different thing.
Yeah. No, I think that's a very good point, because the idea is that the gene expression samples that are out there so far in these atlas-wide projects tend to be from quote-unquote normal, healthy adults. And so the idea is to try and minimize, in some sense, the variation across different samples and use the variation across different regions as the primary axis. So currently, I would say if you're trying to identify sort of causative genetic variations that might be responsible for certain brain-related phenotypes, it's probably not the best idea to use that type of data for that type of question. However, in the future, as we move along, the availability of these gene expression samples that do come from genetically diverse sources is going to open up a whole new set of analyses that can be done to address questions like the one that you just asked. So.
Hi, I missed the very early talk, so it might be a very simple question, but I think it's an important one too, so just make sure. So we know from when relating GWAS or any genes to behavior in psychology or disease, now we know that effect sizes are very, very small in GWAS approach. But there has been sort of hope floating around that maybe in brain imaging the effect sizes are bigger. Has there been any evidence that there are bigger, or what's the replicable effect size in terms of a single SNP influencing brain structure or some kind of functional response to stimuli?
Yeah, go for it. So this is a very interesting question. So I think you can summarize it by saying it's a lot lower than we thought it was, but it's a little bit higher than diseases or cognition. Sometimes, for some regions, in ways that we don't understand. So yeah. So for example, for putamen, we found one SNP that seems to have a pretty high effect size, much higher than those observed for any common variants for schizophrenia. That doesn't necessarily go for other structures, though, that were studied with similar reliability in the same type of analysis. So there are, at least we can say that there are examples where there's a slightly higher effect size for imaging traits than cognition, but it is way, way, way smaller than what has previously been thought, that these will be like detectable with 100 individuals. The associations will be detectable with 100 individuals.
Thank you. We should say this out more loudly, even still.
Any other questions? Yeah. Okay, so just before we let everybody go, there's going to be a evaluation. So please fill out the evaluation. We look at those and we change based on what you say. So that's really, really valued. And second, I just want to thank all the people who came here to tell you about the imaging genetics work. They came from far and wide, so thanks to them very much.